Thank you for the introduction. My name is Masato from Kyushu University, Japan. So first of all, I'd like to express my gratitude to all the staffs and organizers of this conference. So I'm going to talk about genome-first integrated omics approach to complex eye diseases. My financial disclosure is shown on the slide. So today I'm going to talking about two topics shown on the slide. The first is genome-first integrated omics approach, and next I would like to discuss and review future directions and potential applications. Okay, multi-omics analysis can be generally classified into two types: a phenotype-first approach and genome-first approach. In the phenotype-first approach, we compare the molecules such as DNA, RNA, metabolites, and proteins between individuals with and without diseases. And we can identify differently expressed gene or proteins. So in contrast, genome-first approach, we generally utilize the GWAS analysis, the result of genomic analysis, and integrate it with other omics data to interpret it biological findings. So let me explain why I focus on genome-first approach. So as you know, according to the central dogma, our molecule in our body are synthesized based on DNA sequence. So we can find obvious difference in tissue and organs in the affected individuals compared to healthy one, but we cannot recognize the molecular mechanisms. So on the other hand, we can determine differential expressed genes or protein by performing transcriptome analysis or proteome, but we cannot assess its causality because of reverse causation. So on the other hand, germline DNA sequences are not changed by acquired factors such as diseases and environmental factors. So genome-first approach can identify causative molecules and biological mechanisms independent of reverse causation. So as you know, so many eye diseases are influenced by genetic and environmental factors. In case susceptibility is influenced by both genetic and environmental factors, such diseases are called complex diseases. So genome-wide association study is a method to identify genetic loci associated with human phenotypes. As shown on the slide, this analytic method has been applied to multiple eye diseases and identified many genetic loci associated with diseases. However, GWAS has obvious limitation. So I'd like to illustrate the limitation using this slide. So we performed, in recent study, we conducted a genome-wide association study of age-related macular degeneration in Japanese population and identified two new loci. And one of the regional association plot of the identified loci is shown on the right. In this identified locus contains, multiple positional candidate genes, and we cannot determine which genes is responsible for disease onset. So integration with other omics data is essential to narrow down the candidate gene. As you know, omics is a comprehensive study of molecules such as DNA, RNA, metabolites, and proteins. And it is also possible by integrating with DNA data, we can identify genetic locus associated with molecular levels. Genetic locus associated with molecular level is called quantitative trait locus. So if the association signals between GWAS of diseases and QTLs are similar, we can recognize the molecule is associated with disease with causality. So in this slide, I illustrated the analytic procedure of multi-omics analysis in each. First, phenotype-first approach, we evaluated and quantified molecular levels and compared between individuals with disease or without disease, and we can identify genes differentially expressed, gene or proteins differentially expressed, but we cannot assess its causality. So on the other hand, our genome-first approach utilizes the summary statistic data of GBAS and integrate that with other Omics data, and so we can identify causative genes or molecules. I also would like to note that we can integrate GBAS results with other GBAS results. So we perform, by utilizing the association of GBAS results in AMD GBAS, we compare the signals of association between age-related macular degeneration and central serous chorioretinopathy, and found two loci showing similar association signals between these two diseases. This observation indicates that these two diseases share the same biological mechanism of disease onset between these two diseases. Lastly, I'd like to discuss future directions and potential applications. Recent single-cell analysis technology enables evaluation of various omics, including RNA expression, protein levels, chromatin accessibility, and histomodifications. So over the past years, single-cell analysis have been applied to various eye tissues. Here I summarized the studies conducted single-cell analysis across different regions of the eye. So as you can see, a single-cell RNA sequence has been extensively investigated. In contrast, epigenetic states, including chromatin accessibility, have been evaluated only in lens and retina. So therefore, I anticipate that further investigation of epigenetic states and integration with genomic data will provide insight into the pathogenesis of a broad spectrum of eye diseases. So also, recent advances of single-cell analysis can evaluate the expression level while preserving spatial information. So integrating GBAS with this kind of result will further expand our knowledge on ocular eye diseases. Also, I would also like to introduce a new proteome method, OLINK. So this method allows quantification of proteins in a small amount of biological resources, such as tear fluid. Then conducting QTL analysis and integration with GBAS results will further expand our understanding of eye diseases. So now we are conducting a proteome analysis of tear fluids in patients with IgG4-related disease or thyroid eye diseases. Okay, so I'd like to summarize my presentation. So integration of genomic data with multi-omics resources can pinpoint genes associated with diseases. Therefore, genome-first omics analysis can reveal causative disease mechanisms. So also, I mentioned that a single-cell omics analysis and proteome in small amount of liquid material will provide novel pathogenesis of eye diseases. Okay, so I'd like to mention my collaborator, and I'd like to show my appreciation. And this is the last slide. Thank you so much for your attention. And I'd like to note that we are going to organize the 130th annual meeting of the Japanese Ophthalmological Society. And if you have a chance, please join us. Thank you so much.
Any question from the floor? So I have one question, sir. So we always like to integrate multi-omic data set, right? But we know that compared to other diseases like cancer, right? For eye disease, we don't have so many tissues allow us to do the gene expression level or protein level. So what's your solution to these challenges?
Yeah, thank you for raising an important point. agree with the idea, and it's very difficult to investigate the expression level or protein level of eye tissue. So I'm expected that using proteome in small amount of liquids, such as tear or anterior chamber humor or vitreous body, may help to solve this issue.
Any other comment, question? If not, thanks.
Thank you.
Yeah, thank you. Thank you very much. Yeah.